Microplastics are a growing concern faced by environmental scientists and citizens alike. During this webinar, we'll explore quantitative analysis of microplastics in environmental samples using the pyrolysis gas chromatography mass spectrometry technique. I'm Lauren Armstrong, Marketing Director for Quantum Analytics, and I'll be your moderator for today's event. A quick note about Quantum Analytics. We are a value-added distributor of analytical instrumentation in the US and Canada. We offer an array of flexible instrument financing programs combined with technical services like cross-platform installation, training, and product support. For over 30 years, our company has helped labs get the best-in-class instrumentation they need, combined with the services they require to be successful. We offer solutions for a variety of applications, including gas chromatography, liquid chromatography, mass spec, FTIR, XRD, XRF, SIFT-MS, plus a whole range of sample introduction and preparation products, including the pyrolysis technique you're learning about today. We have two presenters for this webinar, Dr. Barbara Schultz-Buttier and Regine Belgena. Barbara studied food chemistry at the University of Münster in Germany and achieved her PhD in 1991 at the University of Braunschweig. Since 1992, she has been a permanent scientist at the Institute for Chemistry and Biology of the Marine Environment at the University of Oldenburg in Germany, where she is head of the Central Organic Analysis Department. She has teaching experiences in organic analytical chemistry, mass spectrometry, coupled techniques, analytical methods in organic chemistry, organic geochemistry, analytical methods in environmental chemistry and plastics in the marine environment. Her research areas include microplastics and additives in marine sediments, microplastics and additives in fish, and definition of baselines and standards for microplastics analyses in European waters as part of the European Baseman Framework. Barbara wrote a number of scientific publications in various journals on microplastic determination and quantification. Her most recent studies in microplastics found in expensive marine table salt, fleur de sel, was widely published in various German newspapers and was even on radio and TV. Regine Belgena is the technical director at Frontier Laboratories in North America. Regine holds a Bachelor of Science in Chemistry and an MBA degree concentrating in strategic marketing. She started her profession as an analytical chemist and worked with various analytical instruments such as pyrolysis, GCMS, TGA, FTIR, and X-rays for material characterization, polymer identification, and contamination analysis. As a former Frontier user, Regine is very passionate about creating awareness about Frontier technology and providing technical support to users. We'll start the presentation with Regine for an overview of the pyrolysis technique, then hand it over to Barbara for a deep dive into the application of analyzing microplastics. If you have a question, please type it into the question button on the bottom right of the screen. We will respond to your questions soon after the webinar concludes. Well, this ends our introduction, so without further delay, I present to you Regine Belgena.
Thank you so much, Lauren, for the introduction. Here is our agenda for today. We start with a summary of who we are, then fundamentals of pyrolysis. After that, we will talk about the multi-mode pyrolyzer, its operational modes, and the method map, as well as sample preparation and system configuration. Then we'll have the application note presented by Dr. Barbara Schultz. So Frontier Labs was founded in 1991 by Dr. Chusan. Chusan, with the support of polymer scientists at Nagoya University in Japan, developed a pyrolyzer based on a vertical microfurnace design. We will discuss the characteristic of microfurnace design shortly. We are a global corporation, and our main products are a series of pyrolyzers, such as the multi-shot and the single-shot pyrolyzers, rapid screening reactors for catalyst analysis, and a line of ultra-alloy stainless steel capillary columns. Our North American Technology Center is located in California. So let's just start with the fundamentals of pyrolysis. What is pyrolysis? By definition, pyrolysis is a thermochemical decomposition of organic compounds at elevated temperatures without the participation of oxygen. Typically, an inert gas such as helium is used. In this technique, a sufficient amount of heat breaks down the organic bonds of a complex mixture into smaller and more stable molecules called the pyrolytes. Then the pyrolytes form and their relative intensities provide insight into the nature of the original composition. It is very important to mention that applying the sufficient amount of heat and identifying the appropriate temperature for programming the pyrolysis furnace are critical factors in pyrolysis technique. As we go more into detail, you'll find out how to identify that sufficient amount of heat so you're not over or underheating your samples. How the multi-mode pyrolyzer can expand the capability of your GC-MS system. As shown here, substances at ambient temperature exist as gas, liquid, or solid, mostly depending on their molecular weight. Materials that are soluble in solvents can be analyzed by GC or LC. Of these compounds, about 30% can be analyzed by GC and about 85% by LC with the proper solvent. However, there are also some insoluble materials that cannot be analyzed using LC, LCMS, GC, or GCMS, but these insoluble compounds can be analyzed by Pyrolysis-GC-MS. In fact, when using Pyrolysis-GC-MS, a solid sample can be analyzed as is, and there is no need for any solvent extraction or sample preparation prior to the analysis. So now the entire range of organic compounds, from gases to solids, can be analyzed by Pyrolysis-GC-MS. And that's really one of the advantages of the multi-mode pyrolyzer and how it can expand the capability of the GC-MS system. So now let's talk about how we can analyze the results obtained from Pyrolysis-GC-MS technique. F-Search is a search engine with 4 unique libraries for identifying additives, volatiles, lighter components, as well as oligomers, heavier than polymeric compounds. It is commonly used in conjunction with the MS library and allows you to also create your own user library to perform contamination analysis, comparison analysis, good versus bad, deformulation, and failure analysis. F-Search has other features like the ability to overlay different chromatograms, extract different M over Z ions, and integrated sum analysis, where the engine can actually integrate the whole chromatogram, make an average mass spectrum, and then identify the corresponding compound. As you could see here, this book is also used along with the F-Search. The book has pyrograms, EGA thermograms, and MS data of major pyrolytes for many polymers. It also has detailed information about the peak assignment tables and EGA. So here is an example page from the book for Pyrolysis-GC-MS of polyethylene. As you could see here, the pyrogram of the polymer, in this case the polyethylene, that was performed at 600 C is shown as number 1. Number 2 category is the peak assignment table together with the molecular weight, relative peak intensity, and the retention index. Number 3 is the EGA thermogram that was performed from 100 to 700 C at a rate of 20 degrees per minute, as well as the average mass spectrum of the EGA. And number 4 category is the mass spectrum of the top 10 major peaks. So F-Search is the search engine software that contains all that information from the book. Okay, now let's talk about the multi-mode pyrolyzer and how it works. As the name says, using the multi-mode pyrolyzer, multiple analyses can be performed on one single sample. This is another advantage of the multi-mode pyrolyzer, where the furnace can be used for other analytical modes in addition to flash pyrolysis. In fact, the multi-mode pyrolyzer can be used for evolved gas analysis, thermal desorption, hard cutting, and reactive pyrolysis in addition to flash pyrolysis. Now let's discuss how to use each of these analytical techniques. We have created a series of techniques referred to as the method map to chemically characterize samples using the multimode pyrolyzer. EGA, which is evolved gas analysis, is the first step, which provides a clear picture about the thermal profile of the sample as well as the optimal temperature zone for the next step. So EGA really guides you what to do next. It will show if a thermal desorption, hard cutting, or flash pyrolysis is needed, or if a combination of this technique is required. To perform EGA, a short and deactivated tube is used to connect the GC injection port to the MS. The sample is dropped into the furnace, which is at a relatively low temperature. The furnace is then programmed to a much higher temperature. Compounds evolve continuously from the sample as the temperature increases. And then a plot of detector response versus furnace temperature is obtained, as you could see in this figure on the right-hand side. So the x-axis is the furnace temperature as well as the retention time, and the y-axis is the intensity. After obtaining the EGA thermograph, you can analyze the peaks using the F-Surge EGA MS library. If it's needed to further analyze the sample, EGA guides you to the next step. You can perform thermal desorption, which is used for analyzing additives and volatiles. However, the key point here is that you are thermally extracting the additives and volatiles from the mixture. So any sample preparation or solvent extraction step is eliminated. You could also perform flash pyrolysis, which is also called single-shot GCMS, and it's a fundamental technique for polymer analysis. In this mode, the furnace is typically programmed at high temperature to break down the polymeric and heavier compounds into smaller fragmentations. The key here is that you are performing a continuous pyrolysis, where the sample is not subjected to heat prior to analysis, as opposed to a pulse mode pyrolysis where the sample is subjected to heat before the analysis starts. You can also do a double shot analysis as shown here that is performing two analyses on one single sample, thermal desorption followed by flash pyrolysis. So first, all the additives, volatiles, and lighter components are thermally extracted from the mixture, and you obtain a thermal desorption chromatogram. And then the flash pyrolysis is performed to analyze the heavier and polymeric compounds from the pyrogram. You could also perform hard cutting, which is the multi-step thermal desorption and pyrolysis. So in this case, the EGA thermogram is sliced into multiple temperature zones depending on the thermal zones obtained from the EGA. This is the flash pyrolysis or single-shot GCMS. As shown here, the EGA was first obtained to identify the optimal pyrolysis temperature, which in this case is 600 C. To perform the flash pyrolysis, the EGA tube is switched to a separation column using the GCMS vent-free adapter. The vent-free adapter allows you to change the columns without venting your MS and waiting for it to be pumped down. So it really saves lots of time and increases efficiency in your lab. Flash pyrolysis generates a pyrogram that is a mixture of all the additives, lighters, and heavier polymeric compounds. And as we mentioned, using the F-Surge additives and polymer libraries, you can now analyze the compounds and pyrolytes from the pyrogram. This is the double shot analysis, which is the thermal extraction of additives and volatiles from the polymer. So first, the EGA is performed. In this case, 100 to 700 C, 20 degrees per minute, and as shown here, the EGA thermogram is obtained. To further analyze the peaks from the EGA tube is switched to the separation column using the vent-free GCMS adapter. Then to analyze the first peak from your EGA, the furnace is programmed from 100 to 320 degrees per minute to obtain the thermal desorption GCMS chromatogram that corresponds to any residual solvents, monomers, additives, volatiles, and lighter components. And then the second shot to analyze the second peak from your EGA is the flash pyrolysis, where the furnace is programmed at 600 C to analyze the heavier and polymeric compounds from the obtained pyrogram. So this is called a double shot analysis because two analyses are performed on one single sample, thermal desorption followed by flash pyrolysis. So now let's talk about hard cutting in more detail. Hard cutting is thermally slicing the EGA thermogram into multiple temperature zones to obtain separate GCMS chromatograms. This mode of operation is a very powerful technique for deformulation and reverse engineering, as well as failure and comparison analysis, since you're able to analyze the composition of the mixture in detail. On the right-hand side, you can see an EGA thermogram of an eyeliner that can be sliced into 5 different zones or peaks, zone A to E. The hard cutting technique allows us to program the pyrolyzer furnace into 5 temperature zones in this case, and obtain separate GCMS chromatograms to identify the components in each zone. So the analysis of each thermal zone yields a chromatogram containing only those compounds evolving from the sample of the selected temperature range. Figure on the left shows each zone and the corresponding identified compounds. Selective sampler shows also on the right-hand side is an accessory that is used for hard cutting technique. From the figure, you can see that selective sampler allows you to selectively pick and choose which thermal zones of your EGA thermogram you are interested to obtain the GCMS chromatogram, and which one or ones you want to vent and not send to the GC column. Sample preparation in pyrolysis GCMS is very straightforward and simple. Depending on your sample type, if it's a liquid or solid, you can use the appropriate sampling tool and place your sample into the sample cup. So samples can be analyzed as is. No need for any sample preparation or solvent extraction prior to the analysis. This slide shows the system configuration for pyrolysis GCMS. One key point that I like to mention here is the fact that the pyrolyzer, which is shown as number 2, is directly connected to the GC injection port. So there is no transfer line that can result in any contamination or losing some of those lighter components. And lastly, let's talk about what sets the frontier microfurnace technology apart from other technologies. In frontier pyrolysis, sample introduction is nearly instantaneous. The sample is placed in the sample cup and is held at near ambient temperature in helium. The microfurnace is then preheated to the desired temperature that is precisely measured with a thermocouple sensor. The sample cup then drops into the quartz pyrolysis tube where the sample is pyrolyzed in less than 20 milliseconds. The pyrolytes are then swept onto the GC analytical column for separation and detection by the MS. So as we mentioned, this technology is really based on a continuous mode analysis, where your sample is held in an ambient temperature while the furnace is preheated. So the sample does not get exposed to any heat prior to the analysis. As a result, there won't be any degradation or evaporation of the lighter components prior to the analysis. That's all I have for today. Thank you so much for your attention. Now I would like to pass it on Dr. Barbara Schultz for her application.
Hello everybody, my name is Barbara Scholz-Böttcher and I'm pleased to talk about the analysis of microplastics, a rising topic we are working on for quite a while. The title is Quantitative Trace Analysis of Microplastics in Environmental Samples using Pyrolysis Gas Chromatography-Mass Spectrometry. Before I start, I would like to introduce our institute. The Institute for Chemistry and Biology of the Marine Environment is located in northwest Germany and has two sites, one in Oldenburg and one in Wilhelmshaven, which is located directly at the seaside. ICBM is host of the research vessel Sonne, and the interdisciplinary structure of the institute consists 17 working groups out of the fields geochemistry and analytics, biology and ecology, and physics and modeling. Back to the analysis of plastics, first a short outline of my presentation. I will start with a short background about microplastics, coming to thermal techniques and microplastic quantification, and then more in detail explain Pyrolysis-GC, giving benchmarking data, examples of environmental samples, and at the end some conclusions. Plastics are very versatile and also very durable. Its anticipated and possible average lifetime is often much longer as its effective lifetime. Over 8.3 billion tons were produced since 1950, and almost 60% ended in landfills and the environment up to now. This plastic waste quite often is mismanaged. Sooner or later it ends up in the ocean and underlies alteration processes as fragmentation. Recent studies show that the smaller plastic particles below 5 mm are the most prominent in ocean surface waters and increase on a logarithmic scale. Here those particles in the range between 1 micrometer and 5 mm are called microplastics, usually subdivided into large microplastics and small microplastics. Microplastics are of raising concern, in particular small particles below 1000 micrometer. This is due to their increasing bioavailability, to their unknown fate, their unknown contents and distributions in the environment, and their unknown ecological effects. And microplastics are an analytical challenge in complex environmental samples. Any visual identification as plastics is not reliable for small particles below 200 micrometers. Microplastics analysis needs a reliable particle identification as microplastic. Common methods are spectroscopical techniques like FTIR or Raman spectroscopy, or fluorescence microscopy, for example, combined with a NIRAD staining. As a result, characteristic spectra are produced to enable a polymer identification, and these techniques are non-destructive. Complementary thermal methods are possible but less common. Thermogravimetric evolved gas analysis combined with gas chromatography-mass spectrometry, Pyrolysis-GC, thermal extraction desorption GC, which is a TGR thermal desorption technique, TGR FTIR coupling, and much more seldom differential thermal analysis or differential scanning calorimetry. These techniques produce characteristic decomposition products that enable a polymer identification, but these methods are all destructive. Spectroscopic techniques are particle-related, and particle numbers related to size and shape are generated. These data are highly relevant for ecotoxicological aspects. But data comparison is hampered. In between samples, the appearance of particles could be very different, resulting in different counts, and particle classification and pre-selection is very variable and belongs to individual selection criteria. And last not least, decreasing particle sizes increase the analysis time, decrease the sample capacity, and increases spectroscopical interferences. Complementary thermal techniques, like for example Pyrolysis-GC, are independent of any particle pre-selection, size or shape. They tolerate inorganic matrix and organic matrix load to a distinct amount. They enable a simultaneous detection, identification and quantification of individual polymer types in one GCMS run, and they generate mass-related data for individual polymers. These mass-related data enable a spatial and temporal study of microplastic content and composition, and a data comparison independent of any particle number or its appearance. The analytical principle of thermal techniques is quite easy. A microplastic particle, here for example polystyrene, is pyrolyzed under oxygen-free conditions. The volatile products are separated on a GC column and result a pyrogram. This pyrogram equals a fingerprint for the respective polymer. Using a mass spectrometer, the detection of single products is possible, and the products can be identified. Here, for example, the styrene monomer and the styrene trimer. The most intensive product gives a high sensitivity for polymer detection. The most chemical significant product, for example the styrene trimer, enables a reliable identification. Extracted as ion chromatograms, intensive fragment ions of these indicator compounds can be used for selective polymer identification, and that even in complex natural mixtures. Using the full scan mode, one can assure the identification of each product via the respective mass spectra. This slide shows the global primary plastic production over the last 60 years. In our method, we include polyethylene, polypropylene, polystyrene, polyvinyl chloride, polyethylene terephthalate, MDI polyurethane, polyamide 6, polymethylene methacrylate, and polycarbonate, since these polymers are representative for more than 80% of the global plastic demand. This slide gives an overview of the polymer-specific pyrolysis products and their indicator ions which were used for identification and quantification. This overview and further details about identification, quantification, recovery, and potential interferences can be found in our publication. The complexity of the resulting polymer pyrograms and sensitivity of the indicator ions can be quite different. Polyethylene, for example, shows a very complex pyrogram, but the signals are still sufficient intense for trace detection. On the other side, polymethyl methacrylate leads to almost one intense product signal. Thermotechniques enable the use of online derivatization. For example, thermochemolysis using tetramethyl ammonium hydroxide as a reagent. Polyethylene terephthalate shows a complex pyrogram with several indicative signals, but with a very poor sensitivity and chromatography after direct pyrolysis. After the addition of tetramethyl ammonium hydroxide for thermochemolysis, the resulting chromatogram shows almost one indicative signal only, resulting in high sensitivity concerning PET detection. Requirements for microplastic quantifications are first polymer standards and an ultra-microbalance, defined and reproducible pyrolytic conditions, and the characteristic indicator ions of specific pyrolysis products. Their signal integration enables a calibration. Quantification with pyrolysis require a very high reproducibility of pyrolytic conditions. In our first studies, we used the Kyripoint pyrolyzer that enables a very precise and reproducible pyrolysis temperature due to a defined ferromagnetic conductor. And it has very short pyrolysis times. The capacity as well as the dimensions of the pyrolytic cups are quite small. The microfurnace pyrolyzer bases on a very precise programmable ceramic heater with high temperature stability. Due to a multi-shot option, preceding EGA measurements are possible. The capacity of the sample cups is clearly bigger compared to the Kyripoint pyrolyzer. This slide compares the calibration between both systems for 3 polymers. The Kyripoint system allows a calibration up to around 1 milligram of a single polymer, but it shows very high process standard deviations. The microfurnace system, the maximum of a single polymer is much less. Weights much higher than 50 microgram leads to saturation effects in some cases and should be avoided. The use of an internal pyrolysis standard further reduces the process standard deviation and enables quantification even much below 1 microgram on a semi-quantitative basis. The limit of quantification and of detection for this approach is highly restricted to the balance used. For hardly solvable polymers, shown here for polypropylene as an example, the LOQ and the LODs estimated using the calibration function only are around 1 microgram. But regarding the signal to noise ratio, 1 microgram is not the detection or quantification limit, here shown as an example for polyethylene terephthalate. Working with solvable polymers, much lower limits are available, as shown for polystyrene. And here also the combination of solved standard and standards using the balance is shown quite nicely. Looking at the lowest calibration end, LODs and LOQs in the nanogram range are visible for the styrene trimer and even much lower for the polystyrene monomer, where the limit of detection is 3 nanograms. Overall, the method shows a high potential for trace identification and trace quantification of polymers in a nanogram range. Coming now to environmental samples, from literature, particle-related numbers of microplastics are available that are spanning a broad range and are given in variable dimensions. In some, a low microplastic content is often accompanied by a high organic and inorganic load. This makes an adequate microplastic pre-concentration mandatory, which leads to an effective removal of organic and inorganic matrices and does not affect the polymers. Overall, the bottleneck of Pyrolysis-GC is a small pyrolysis cup. Sample-dependent, we use a combination of different methods for microplastic extraction from environmental samples, such as sieving cascades, for example for water samples, density separation with high-density sodium bromide solution for sediments, enzymatic chemical oxidative digestion for biota samples and sample-dependent for others, oxidation with Fenton reagent, and in some cases a combination of all. At the end, the whole sample residue is filtered on an anodisc or glass fiber filter of a very small diameter that is directly placed into the pyrolysis cup. Obligate to evaluate the data quality is to run a consequent procedure blank along the whole sampling and cleanup process due to an immense risk of secondary contamination. And now to the presentation of some results. In a pilot study, we analyzed the stomach and gastrointestinal tracts of fishes from the North and Baltic Sea. Here an example of a flounder is shown with the total ion chromatogram and the ion chromatograms of selected indicator ions of respective polymers. In all cases, the sample is shown, a polymer standard is shown, and the procedural blank. In this case, the flounder sample was positive for polyamide, for polyethylene terephthalate, for MDI polyurethane, and for polyethylene. Around 70% of the fishes were positive for microplastics in their gastrointestinal tracts, and the microplastic concentration was in ppm range. This example was performed with chiral-point pyrolysis, and therefore the concentrations are at the lowest end of the calibration curve. The next example shows the results for a typical muddy Wadden Sea sediment analyzed with microfurnace pyrolysis. The total ion chromatogram is shown in contrast to selected indicator ions of PMMA, PET, and polyethylene. In total, 6 different polymers were analyzed. The sediment was analyzed as triplicate, and the differences in abundance represent the variability in natural particle distribution, which seems to be much less for polyethylene compared to the other polymers. All in all, the concentrations found in these sediments are in ppb range. The third example shows the results of a successive filtration of 220-liter North Sea water. The microplastic content shown for this volume is very small and in the range of quantification limits. But however, 3 different microplastic types are detected, and the 2 fractions, bigger than 20 micrometers and bigger than 125 micrometers, show differences, which have to be discussed. The overall observed concentration of the sample series analyzed range between PPT and ppb levels. As an almost natural product with a direct marine context, we studied fleur de sel from the Atlantic Ocean and Mediterranean Sea. Here is an example shown from the Greek. Besides several brine shrimps, or better that cysts, and relicts of brine flies, plastic particles were visible even under the microscope. The quantified microplastic content was in the upper ppb to ppm range. The plastic types detected were very closely related to typical short-term packaging plastics. The analysis was performed 5 times, and the samples were directly analyzed without further homogenization. This was done to avoid a particle fractionation in the sample and is clearly shown here in the variability of the data. This slide gives an impression of the spatial fate and distribution of microplastics generated from data of the fleur de sel study. Without further stressing this figure, it shows the potential of the generated data for further discussion regarding sources and concentrations of microplastic in the environment. To wrap up the presentation, we have seen that Pyrolysis-GC is complementary to FTIR and Raman techniques. It enables a selective, simultaneous, and sensitive identification and quantification of common plastic types in complex environmental samples within one single GCMS run of the entire sample. It based on characteristic indicator ions of polymer-specific pyrolysis products. It was presented for 9 polymers, and this could be further extended, even retrospectively. The analysis is independent of matrix residues to a certain extent, but microplastic pre-concentration from environmental samples is usually required, and we get mass-related microplastic quantification on a trace level. These data enable a temporal and spatial study of microplastic load in variable environmental samples of different origin, related to masses, composition, and distribution of these microplastics, and that independent of its appearance in terms of shape, size, number, or color. Here we have seen mass-related microplastic data in environmental samples for the first time for surface waters in the PPT range, sediments in the ppb range, biota in ppm range, and in case of the sea's marine salts in pph to promille range. At this point, a data synchronization between FTIR and Raman techniques and thermal methods are necessary. This is already in progress. So for identification, this is done comparing micro-RT-FTIR spectroscopy and pyrolysis-GC. And for quantification, this is in preparation, in this case for FPA, micro-FTIR, and pyrolysis-GC. At the end of my presentation, I would like to highly acknowledge the work of our microplastic team, namely Martin Fischer, Moritz Halbach, Anke Mühlenmeister-Sawal, Christopher Dipke, and Isabel Großmann, and the German Ministry of Education and Research for funding. Surely our working group, and that's for sure. Thank you for listening, and I hope you have a lot of questions now.